Hello and welcome to this video, which is going to be a bit of a revision session on basic bacterial structure. We'll consider a few different angles, what the bacterial cell is made of, one or two classification steps and ways to classify bacteria, and also we'll relate it all back to antibiotics. So I'll try to relate the structure back to some of the drugs that you'll prescribe in the future. On the screen, I've just got a fairly generic image, and what we'll do is I'll swap the camera around in a minute, and we'll go through and we'll draw through a picture of a bacterium. Again, you can kind of do this in your own time. There's a few specific questions on the screen here. So, how do bacteria differ from eukaryotes? What surrounds and contains the cell? How do bacteria respire? Bacterial DNA, and if we were to think about antibiotics, where might they work? So what we'll do is we'll start by just swapping the camera around, and what you can see hopefully on the screen is the screen. You can see my hand here, and we're going to start with a fairly generic egg-shaped thing. We'll consider a few things. So we'll consider morphology. We'll consider respiration. And then we'll consider the cell structure as well. The cell is in a kind of, I suppose, rod-shaped or bacillus. So we've got an outline of the cell. For the sake of argument, what we'll say is that this is the cell wall. Now, the cell wall of the bacterium is comprised of different elements to it, and specifically within that, we will say there is the molecule peptidoglycan. This is, as the name suggests, it's a peptide linking with a glycan, i.e. with a sugar. You have alternating sugars, which are called N-acetyl uramic acid, and N-acetyl glucosamine. NAM and NAG. And these are sugars which are exported outside of the cell, and they are linked together to other chains via a peptide linker. And the link here is created and is generated by an enzyme called the transpeptidase. And this is an enzyme which is an antibiotic target. So we've got an outline to the cell wall. Now we also have, on the inside, running all the way around, we have a cell membrane. Now, this is always present. What I mean by that, as a flip side, is that the cell wall is often, usually present, but not all bacteria have a cell wall. The cell membrane is always there. The cell membrane is a lipid bilayer in which we find, along the way, we'll find the respiratory complex. So we'll find bacterial respiration takes place here. Okay. And I'll just draw a few boxes. These boxes represent what is an enormous molecular machine embedded within the cell wall. But I'll just put down here respiratory complex. So we've got a cell wall, we've got a cell membrane. What else? So we'll have, in addition, we'll have a chromosome. Now I'm going to draw this as just essentially just a kind of a squiggly line radiating out from the central point. Now, bacteria have one chromosome. Now, this is sometimes also referred to as a nucleoid. And as the name kind of suggests, and as you would find in your own body, this is made up of a double-stranded DNA molecule. It's coiled around, and there are various proteins and ways bacteria will coil that and package it efficiently, but they don't have histone proteins like you would expect to find in a eukaryote, nor is it bound within a nucleus. So this is one way in which the bacterial cell differs from your own cell, i.e. the difference between a prokaryote and a eukaryote. In addition, we'll have plasmids. And plasmids are essentially parasitic bits of DNA. These are essentially, they vary from a few thousand base pairs to maybe several 100,000 base pairs. And they are parasitic bits of DNA which will utilize the bacterial cell to replicate. They will utilize host cell nucleotides, but they often carry useful genes for the bacterial cell, which creates a selection pressure for the cell to retain the plasmids. The plasmids also themselves will sometimes force the cell to produce a pilus, which allows them to move from one cell to the next. And that allows us, in saying that, to think about the next structure. A pilus, or more, or to say a pili, which is the plural, are essentially protein tubes through which plasmids can move. Pili can also move backwards and forwards, and they also have a role in attachment. And the attachment aspect is also important when we consider another broader class of molecule called adhesins. And these are cell surface structures, glycoproteins, proteins, some other molecules, so molecules like LTA, for example, lipoteichoic acid, found in gram-positives, of course. These are the things which also can allow the cell to attach to surfaces. What else do we have? Well, we have ribosomes. Remember, a ribosome has a small and a large subunit. And the large is of a 50S and the small is a 30S. These are not quite, these numbers don't quite equate to the mass of the ribosome, but you can kind of take it to mean that these are just the units that the size of the ribosome is measured. It's a different conversation, and it's arguably less important. The key thing is to say that the size of the ribosome is different to that of eukaryotes. So what other questions do we need to consider? So we've got, how do they differ from eukaryotes? So we've got the chromosome is not bound. You'll note that the respiratory complex here, this is the site of ATP generation. So bacteria don't have mitochondria. They have a cell membrane, that's always present. They also have a cell wall. That's a key part of one of the main differences between your cells and bacteria, and that's an essential drug target. And you also have ribosomes. Now, of course, your cells also have ribosomes, but the sizes are different. There are other things going on inside as well, and I suppose what I'll just do is I'll just label these as bacteria-specific enzymes. It's quite a horribly generic and broad term, but bacteria are capable of catabolic and anabolic functions that your own cells can't do. That means that they can build molecules, they can synthesize things that you can't make, and they can sometimes break things down as well. So certain plant metabolites, for example, that can't be utilized by your own cells can be broken down by bacteria. And there are specific examples here which can be inhibited as for potential therapies. While we're thinking about that, let's move on and think a little bit about potential therapies. So just use a different colour. Well, ribosomes. Ribosomes are an important drug target. We have antibiotics such as macrolides, which work at the 50S subunit. Macrolides are a useful class to remember as alternatives for penicillin. We have antibiotics that work at the 30S, so these can include tetracyclines. They can also include the aminoglycosides. What about cell wall? Well, we're going to consider here arguably one of the most important beta-lactams. And the beta-lactams can be further subdivided to penicillins, carbapenems, and cephalosporins. There's a fourth group, but just for brevity, I'm not going to include that fourth group called monobactams as well. We've got other groups down here. So we're going to also include glycopeptides. And I'm going to put this on at a slight angle because I have misjudged where the edge of the paper is. The glycopeptides are essentially involved in disrupting the synthesis of these building blocks. So what basically happens is that these molecules become, well, it just becomes harder for the bacterial cell to build itself and work in a slightly different way to the beta-lactams. We also have cell membrane. Now this is a rarer drug target, but there is a couple of, or one specific antibiotic that's quite important to note. Now, actually I spelled that wrong. Polymyxins, and also another one which is called daptomycin. The cell membrane is a rarer drug target. The challenge with it is how do you selectively kill the bacterial cell, disrupt bacterial cell membrane, whilst leaving your own cell membrane alone. Respiratory complexes, well, again, these are kind of harder to target, but we'll say here, one of the drugs which targets the respiratory complex is metronidazole. And this is a useful antibiotic for treating anaerobes. And then lastly, we come on to double-stranded DNA. Two useful groups here are the enzyme inhibitors so these can include quinolones, and also I've spelled that wrong. Fluoroquinolones also fall in here. Fluoroquinolones are, or were arguably more commonly used, but their use has been is under quite a lot of review because they have quite some quite nasty side effects. Nevertheless, they're still quite a useful antibiotic group to be mindful of. So examples like ciprofloxacin, for example. The other one to note is trimethoprim. And this is a folate antagonist. Folate is a building block for DNA. So you starve the cell of making folate, and then it interrupts or prevents the cell from replicating itself. So you've got a few different groups targeting different structures and different processes going on within the bacterial cell itself. It's a good idea just to become a little bit familiar with these things. And it's important when we start thinking about how antibiotics work. As future prescribers, you are going to need to know how they work. And what about morphology? There are a few key terms to note. So you can have bunches of cells together. These can be staphylococci. You can have chains of cells. These are strep. You can have individual cells when they're just kind of growing individually. These are just the cocci. Just bring that over. Sometimes you'll have cells joined just as pairs, and these can be the diplococci. Rods look a little bit like this. You may sometimes see, just a kind of slightly different scale, what looks like a slightly, I don't know, a slightly elongated coccus or a slightly shorter rod, depending on your I don't know, depending on how you look at it. And these can be what are called coccobacilli. Haemophilus is an example of one of these things. So a range of different morphology, and it's not exhaustive, by the way. So you have spiral cells, so spirochetes, and there's a few other things to note. And what about respiration? Well, the key thing, I suppose, is to note that we have both aerobes anaerobes, and then those in the middle that can do a bit of both. And these are facultative anaerobes. All of these things, it's a little bit of a lot of busy stuff going on here, but all of these things are useful points to pick up on, and we can come back in future videos just to pick up and go into just a tiny bit more detail. But from a first year point of view, I hope that was useful.